Olá, meu nome é Michele Letícia Macan, sou residente nível 2 de Clínica Cirúrgica de Pequenos Animais da UFPR Curitiba. Eu entrevistei o Dr. Maximilian Stamnitz no grupo de pesquisa de tumores transmissíveis da Universidade de Cambridge sobre o tumor facial transmissível do diabo da Tasmânia, como parte da avaliação da disciplina de Seminários.
And what areas in Tasmania are actually most affected, and how did it all started? Like, whereabouts did it start in the DFT1 and DFT2?
DFT1 was first observed, as you said, in 1996, and that was in the very northeast of Tasmania. It's a national park called Mount William. And there, at that time, there was a very dense population of animals. Some people think that we've never seen more Tasmanian devils than in the mid-90s. So there were a lot of wildlife photographers back then, and a guy from the Netherlands called Chris Tovars, he did like night captures of devils for a long time already, until on that particular day, he just photographed a few animals which had these tumors.
Okay.
So it was at first a very local phenomenon. And from my genetic analysis, we know that we could reconstruct basically how closely related the individual was that had the tumor, the first tumor, how closely related it was with populations around the island. And through that analysis, we basically also found that it was closest related with animals in the Northeast. And from there, it then spread very fast, both southwards and westwards.
Mm.
And at the moment, we think that the only, or maybe it's already past that point, we think that the only areas where DFTD has not yet affected animals are the remote northwest and remote southwest. But as far as I know, all the cases that we've seen so far are just in this small stripe of land. So it's a much more recent phenomenon and a much more local one than DFT1. So the conservation problem for the devils at the moment, it's really just DFT1. More precise, and there's modern genetic ways to refine that picture and trying to understand better how this particular type of cell could be more vulnerable than maybe a lung cell or a cell from the pancreas. I mean, it could be any cell that gets a cancer, but maybe there's something special about these types of Schwann cells to be transmissible also. So
I was actually wondering, is this tumor actually like aggressive only locally, or does it actually spread throughout the body and metastasize, or is it just like on the usually on the skin cells, or does it have a predilection?
We know that it can metastasize for sure. So people, well, not so much anymore, but in the early days of DFTD, there were a lot of autopsies being done. You found a dead animal on the road or somewhere in the woods with a tumor in the face, and then like the vets cut it open, and you do find every now and then that there's distal like lymph node infiltration of these tumor cells, for example. And sometimes metastases in the lung or like other organs. We don't know for sure how frequent it is, but we know it can happen. I know that there has been some controversy about that. Like I think in the community, the most supported idea is that it's direct transmission only.
Okay.
But I know sure that there's at least one very prominent ecologist who still at times argues that maybe, it could be spread through the same food source.
Oh, yeah.
Like an animal biting a cadaver and then another one later picking up tumor cells through the same cadaver.
Oh, yeah.
Certain part in the face or in the skin, which, suppresses the growth for some time, but then some sort of switch happens and the tumor starts spreading there. Which, from pathogens point of view, it's a bit like with COVID actually.
Yeah.
if it were like, to be without a latency period, this might actually be very dangerous, because if it grows so fast, then it could be that many animals die before they can transmit the disease, and then the disease would thereby not be so efficient.
It transmits really easily between individuals, doesn't it?
Well, actually, it's hard to say,
Yeah.
because it's not easy to do like experiments of this sort. Like you wouldn't get an ethical approval for good reasons.
Yeah, definitely.
Put like an infected animal and a healthy one in the same cage. You're not allowed to do that.
Yeah.
So it's hard to like follow the life history of a single infected individual and see how often it infects others.
Or even a healthy one and see when it gets infected. So
yeah, exactly. So in Tasmania, there's some ecologists which try to, like, Rodrigo Hamidi and David Hamilton. What they're trying to do is to study a population with radio collars. So they basically label these animals, and they can register like how often these animals contact with others. So whenever like is close to another, it will trigger a signal. And with this sort of analysis, you can try to understand what happens, like how often do they meet in general, and how can that change when they get sick, and how likely is it to make others sick if they meet them. But for example, I think you asked about that as well, the latency period.
Oh yeah.
We don't really know it because we don't have much experimental data. But we know of a few cases, where an animal was observed more or less healthy, then isolated in a zoo, and then several months later developed the disease. Okay. So they can be asymptomatic for like a substantial amount of time. That we know for sure. But the question is if that is happening often, or if that is just like some individuals with maybe better immunity, maybe just less inoculation, fewer cells that start the growth.
I see.
All of that is not so well characterized. But what people tried in the early 2000s, mid 2000s, was to close off an entire peninsula forest here in Tasmania. And they basically tried to eradicate the disease by killing every single animal that they found to have at least a tiny tumor.
Oh wow.
So the idea, if it's growing very fast, then we could just wipe out the disease by like taking out all the infected animals. And what they found was that this actually didn't work.
Oh, that's a shame.
Yeah. And maybe it was because they just didn't catch all of the sick ones, all the visibly sick ones.
Yeah.
But much more likely, I think, is that there are some animals which had the disease, but it wasn't visible for too long. And then, like after a few months, when you come back and you think, maybe this place is sort of safe now, and you still saw the same number of animals. As humans, we have a huge impact on Tasmanian devil populations by killing them with cars, by not managing forests in a favorable way for them. So nowadays, it's not just DFTD that's a big problem, and for that reason, even if evolution happens, it might not be enough to save the animal. But there's a big controversy around how the best conservation would be done. Some people say maybe we should just leave these animals alone and let them become resistant, and others say no, we should actively breed animals, bring them back into the areas to repopulate areas where many devils have died out. But these animals that we breed, they might not be resistant, so maybe it's not, makes no sense. So there's a big community discourse around this subject.
In a very early stage, I would say, still like to get all the answers to how the disease actually works. So there is a lot of effort that's been put in, and there is a lot of things that still need to be investigated. That's what makes it interesting as well. So
Exactly. Yeah, I fully agree with you. I think it's worth putting more resources into this type of work, because like you don't only learn about Tasmanian devils and their cancer, you learn about, general ways in which pathogens can start, spreading, and might be able to apply knowledge from this, like for human cancer research. So yeah, it's a very extravagant type of phenomenon in nature. Muito trabalho, né?
Definitivamente.